So what exactly happens is, okay, so the clinical trial closes, okay, fine, clinical trial gets closes. The study is locked, the study ends. Now what happens is that, now this is a situation where we start with the clinical SAS coding. Okay, what happens is that there is a PM, project manager. This project manager has, suppose, is looking over this particular clinical trial. Okay, so then the first thing that he allots is uh, the raw data. Raw, he looks into some of the things like raw data synchronization. Correct. So remember when I told you that all the data which comes here, which actually is collected in the EDC system, the raw data is also structured and shared in different tables based on their domains a little bit. Remember, because that raw data will be the data set, will be ultimately, those raw data will be represented in SAS data sets, and from those raw data, the SDTM, ADAM, and TLM, and so on and so forth will be going here. We will be carrying out. The team will basically be carrying out. Correct? Yes, no?
Yes. Yes,
Raw
yes,
data,
yeah.
yeah, okay. Raw data synchronization in SAS data sets. Okay, so these are basically the activity that he is looking into. So suppose 10 people are there. So there is there, so 10 people will work on it, and this person will define timelines. Okay. Second, what we have, then we have SDTM specification. Okay. Before this SDTM specification, there would be annotation, annotation of CRFs. Okay. So annotation of CRFs is what? You will aligning the CRF questions to see this standard elements. Okay, what does that mean? That means you have the questions which are there. Remember the annotative CRF that we had shown, where we discussed about. Besides every questions, we have those standard series questions which are there. So if it is a name, if in the CRF it is given adverse event term, then against that the series term which would be aligned would be AE term from the AE domain. Again, A is, suppose we have a question, adverse event start date, adverse event end date, adverse event severity. So these are questions which are asked. But these are responses will be standardized, and where will they be fitted in the SDTM domain? They will be fitted in AE term, AESTDTC, adverse event start date, AEENDTC, adverse event end date, AEACV, adverse event severity. That is something which will be done by a team. Again, suppose it will have 10 people and the timelines are given. Okay. The timelines are given for timelines for development and validation. There will be a team who would be doing it, and there would be a validation team also who would be checking it, reviewing it, whatever. There would be a team. Once this is done, then we do a go ahead with SDTM specification. So based on this, the SDTM specification is created. Okay. So SDTM specification is, so for DM domain, for every domain, this specification is created. So for DM domain, what do you check? If I go here to the DM domain, suppose, uh, yeah. So So study ID, domain ID, all these things are written. So how it will be derived? Like study ID, this will be direct. Domain should be DM domain. So U subject ID is what? It would be a concatenation of study ID, site ID, and subject number. So we have this study ID, site ID, and subject number in the one of the raw data sets, raw STTM data, I mean raw SAS data sets, which is where, which is actually this one. Okay. Then what we have after SDTM, we have what is the RFSTDTC. What is RFSTDTC? When the person first took the drug. What is it? Set minimum of ex.exdat. So it's given how you will get it. So minimum of ex.exdat means what? There is an ex raw data. From that you take this column, concatenate with this column which is also present in the ex data and in ISO format. So these are the derivations which actually based this. There is a team who does this specification, creates this specification based on what? Based on the series SDTM IG which I had shown you, that PDF, and also with that annotation of CRS. How that annotation is done. So this SDTM specification is created. Suppose this has 20 people. Okay, and this has the timelines for development and validation. All these the project manager is looking into, correct? One after the other. Once this is done, what do we do? SDTM specification is done, then creation of yes, then you can say Adam specification, correct? Okay, let's do Adam. So this can be done by a separate team. This will be done by a separate team. So we can go parallel, very good. Sometime in startup, they are done by the same team. Okay. So Adam specification, suppose there are 20 people. This is the same thing. Cool. Now we have SDTM creation. Now we create the SAS datasets for SDTM standardization. Okay. Fine. So now for that, we have suppose 10 people. 10 people are assigned. Again, timelines for development and validation. Same. So let me do one thing. Let me wrap this text a bit. Okay. SDTM creation, 10 people, timelines for development. Then we have Adam creation. Okay. Same, suppose give 10 people are there, and this is the timelines, and the person has timelines for development. So every 10 people, so Adam, this person will have all the Adam datasets which are needs to be created. List in the same Excel. All the list of Adam datasets that would be there, and the timelines, when will it be take, when it will not be take. That same for SDTM, same for Adam. Then once this is done, all the review and everything is finalized and locked, then comes the listings. Listings, tables. I give here 20, 20, 20. Okay. So this will be same thing. And this is where you end your study. This is where you get an entire data created of everything. And this will be, the entire thing will be handled by the project manager. Okay. Project manager will look into this, all these things, whether they are happening correctly or not. So you will be placed somewhere here, or somewhere in all these things, somewhere. Or you might be placed in Adam SDTM creation, or basically Adam listing tables creation, anywhere. So there are specific teams who are there who will be working, and the project manager will be looking into this entire timelines and what is the first problem. There also will be a program manager. So project manager will be looking into the timeline and everything. Program manager will be there who looks into the overall. He has knowledge of everything. He will also look into everything, a senior program manager. Like what is the problem, what is happening, why do you need support from the client, what not. And the project manager will also deal with the client with the timelines where we need some help and whatnot. And all these 10 people will consist of, suppose, 8 junior members, 2 seniors, 8 juniors, 2 seniors, who would actually help them to navigate through these things by creating this, their SDTM annotation, SDTM specification if they get stuck. This is the entire workflow. Got it?
So yes, yes. So how like so almost 100 peoples in a one team.
Yes. If it's this is 100 people in one team, but there are, suppose, 2, 3 clinical trials that they are handling simultaneously.
Okay. So if I am working over there, so I can handle two, three project at a time.
You have to handle 2, 3 projects at a time.
Okay.
It's not necessary, but because I'll tell you one thing, you will be working for 9 hours.
Yeah.
If it is a difficult, because people will be discussing, like if you join at a bit senior, they will ask you, Acha Priyanka or Shagufta, tell me that these are the 5, 10 listings which we are giving it to your team. How much time will it take? You, based on your understanding, based on your team's knowledge, you will say that okay, these are 5 ones. I mean, for one trial, we are thinking of assigning 5 trials. How much time it will take? So you might say that okay, this will take around. So each listing would take, if I give distribute it among 3 people, it will take among 2 days or 3 days. Then there are still two resources who are pending, who are there, 2 people. So they will say that your senior will say that okay, there's another trial. I'll give 5 listing. You can distribute among them. That's the thing. Because you're working 9 hours. Suppose one listing you take 3 hours, and the rest 6 hours will you be sitting idle? No, you will be working on something different. That is a separate study which you will be working. Similarly with the validation people, there will be another team, a validation team as well, separately, who would be working on the same things which you are working. Yeah. So there's a separate validation team who will work on this. Then the development team and validation team will see, okay, is the SDTM specification, not specification, but is the SDTMs which are created, both are in sync? If not, what is the problem? Why they are not in sync? What was missed to understand? That is where happens internal validation. Then Adam validation, then listing, table, figure validation. Like we are doing here, the table, the listings which we are seeing here, same thing. Suppose Shagupta is also doing same listing. Now Shagupta comes in with her output, I go with my output, and then we do a proc compare. If we see that there are differences in numbers, what are the differences in numbers? What has Shagupta captured which I have not captured? Or what has Shagupta taken into consideration which I have not? Now, if there are differences in values, how come the differences in values coming? What is the problem? So these are the validation. I'll just develop it, go and submit. That will not happen. There will be an internal validation. We will look into it. Then we will submit it to the client for their validation, final validation. Got it? Clear?
Yes. So suppose I am working in the SDTM creation. So if it is the member of, like there are the 10 members, right? So
Hmm.
in that there are some junior, senior, and team lead. It will be
It will be like
like that. It will be like okay.
correct. And if it's 2, 3 clean projects, then in the SDTM creation team, there would be around 15 people who are working, suppose.
Okay. So like a team lead will be like two or
Two team
one.
leads are there, who is looking into that. Then there are some senior resources. Then there would be some junior resources, and there would be some freshers or staff people who would be working. That
Okay.
is
So
exactly the hierarchy that happens. Yeah. So,
okay. So where is the biostatistician will be there? Like in this where he
biostatistician,
works, yeah.
biostatistician basically comes in this table part when they come, but that is mainly from the client side. From the client end, the biostatistician will be there. From the from EY, I mean, so from your organization side also there would be a biostatistician, but that comes mainly in this part while developing the efficacy tables and the figure values. That is where they come in. Who has bias, but they will not exactly have the SAS knowledge. They will have the statistics knowledge. This is something, the tables and figures which you would be creating, you would have, they expect you to have the SAS knowledge. They will see that the value they have manually derived from the data and the way you have derived using SAS, are they matching or not? The biostatistician is not bothered about SAS program. Okay, he or she will get the data with using Excel or using, if he knows SAS or using anything, he or she will try to get the value of the efficacy, suppose, or the value of the 95% interval. All these values he will get, and the different tables, converting that thing into SAS program is your responsibility because you know SAS. The biostatistician doesn't know. Biostatistician doesn't have a SAS background. He has a statistics background. So this biostatistician from your organization and that biostatistician will be conversing with each other, and this the biostatistician in your organization will rely on you a little bit to work on the SAS program. Now you work on the SAS program. If the values doesn't match, then they will ask you to revisit what exactly has happened, what are the values that you have taken, why is it not coming up, is it the correct SAS programming that you have taken. That is where the complexity lies. So people who has bit more experience are given these efficacy tables. Got it?
Yeah. So one or two biostatistician, like how many,
Yes,
like,
yes, one or two maximum for each, for 2 3 studies. I have seen like the amount of biostatistician is less. See, this is a major biostatistician is someone who actually designs the SAP, the efficacy tables, like how the efficacy procedure, how, I mean, how the efficacy should be calculated, what procedures should be used to calculate, what are the statistical methods should be. So they from a high level, so they are basically PhDs who in a clinical trial they give their inputs in this efficacy calculation. They are not bothered about SAS program. They are bothered about the value that is coming in. Is the value correct? Is there the idea that they have as a biostatistician, the value that they have got? Because if you have the data, suppose all this patient data you are is there in an Excel. Those data they will take, they will do some Excel formula calculations, and after that they can create the. I mean, they will get the efficacy value. They will see, but that is something which you can't submit to FDA. Correct?
yeah,
You have to submit it in a beautiful table format. You have to have SAS program. So SAS is, that's why statistical analysis system. It allows you to do statistical analysis. So there would be in the SAP mention what are the statistical procedures you will use, and based on that you will have the SAS program written in that way. That idea will come slowly, and based on that method or based on that program, you can get the statistical value in a good perfect format in SAS for that particular table. But the value is, when the value which is coming, that is where the biostatistician is interested. Is it coming correctly as per the expectation of his or her? Correct.
yeah,
But the problem is, why is there more competition in biostatistics? Because for 2 3 clinical trials, there is only one or two biostatisticians. But there are so many tables and so many things that needs to be done. That's why clinical SAS programmer is required in so large numbers, with experience, with juniors, with freshers. Like, have you seen very frequent biostatistician openings?
yeah, no,
Exactly, you won't, because they are very high level PhD people who have been into statistics for around 20 25 years. They are the people who design this stuff from a statistical point of view, and that we need 2 3 people for around 6 7 studies to design it. Same with data scientists. How many data scientists we need from work with it? We need one or two. But once this model is described or model is finalized, then to implement it, to make it integration, we need more data engineers. So data engineers is in lot demand than data scientists, because data scientists work, I mean, with one or two data scientists, you can achieve a lot. With one or two biostatisticians, you can achieve a lot. But all these things, 2 3 clinical SAS programmers won't be able to do it. So many SDTM specifications, so many add-ons, SDTM. That is where they are required in bulk. Got it?
Hi, Archit. I have a question.
Go ahead.
So the biostatisticians are the ones who coin and tell us like these are the procedures that needs to be done, the statistical methods that are supposed to be performed. But we have to know like how to do
Uh,
it programmatically, right?
in SAS, yes. And for that,
So yeah,
yeah, go ahead.
we haven't done any of that type
But
of, analysis until now.
but I have couple of tables with me, which I can give you couple more from another project. But that's it. I also don't have. I've not also worked on that extensive efficacy tables. You won't find people so much like who has worked on extensive efficacy tables, and they. I mean, you will find people, but they are not at our level. They are people who you are. If you have, if you are working in this SAS program project for suppose 8 9 years only in SAS programming, clinical trials, clinical SAS, then you get a chance to work on this. But don't get bogged down by efficacy tables. That is not the only thing that they are doing. So there will be people, and on the process you will learn. Okay, and it's very difficult to get efficacy tables and get everything. I mean, how you are doing the efficacy tables, what are the so data getting data in hands-on is also difficult, right? That's what I'm trying to make you understand. We have couple of tables. We can do that, but the rest you can do some. You can just do a YouTube and you can just do some Google search and get an idea of that. So that will be enough because I
So another question that
uh,
I have is
uh,
like, what's the difference between the NDA and ANDA, clinical part? Because a lot of things, I mean, more half of the things are excluded in ANDA because we're just trying to compare it to a drug that is already in market, right?
NDA is new drug application which goes to the FDA.
ANDA. What's the difference between the clinical reporting part of ANDA and NDA? What is excluded from ANDA that is included in NDA? Like the tables and listings, SDTM, any difference between
ANDA and NDA. No, I'm not clear about that. Let me get back to you. Let me see what exactly you're saying. Difference between NDA and ANDA.
ANDA? Yes, abbreviated new drug application.
Okay.
It's a generic drug that is being compared to a branded one that is already in market. And yeah, that's how mostly only the bioavailability is carried out for those drugs. Even like clinical trials are also done for them, and clinical reports have to be submitted in order to get the approval. So I wanted to understand like what's not included in those reports.
Okay, then I will check with it then. Then I need to check for it once.
Okay, thank you.
I can check and get back. Yes. So I have a friend of mine also who's working in that. Like, she can also help. Okay. Let me just ask her and let me also take a look myself and I'll get back. Any other questions? Any other thoughts that you have?
So, so here, where we are using the Pinnacle 21 software.
This part.
So after the specific, okay, after creation of SDDM and ADAM,
Is
right?
have you created SDTM and ADAM correctly
Okay.
as per the CDIC specification and everything? This is where you will be creating using Maker 21.
Okay. So we have to create this like data, like this SDDM and ADAM data set in XPT file, right? For that.
No, SDTM and ADAM dataset you'll create in SAS. You will export it out of SAS in XPT. That is where I mean, when you export your SAS, when you export this out of out of SAS, it becomes an XPT format. That is a format that you will upload in Pinnacle and then select which SDTM IG version you have used to create this. Is it 3.2, 3.3, 3.1? And after that, you run it. Once you run, if there are no errors will come, and if there are errors, the findings will come.
Can you show like once we create this, if you have the like how to upload and how to create
I
this?
don't have Pinnacle 21 here.
Oh,
In here, I don't have.
okay.
But I can show you the XPT files. Let me see if I have the XPT files. 1 second. I will have the XPT files. XPT files, right? Ah, dot XPT, a dot XPT. See, I don't know if I double
So
click if it will come or not. Let's see. No, it won't come like this. You have to upload. You have to take it up somehow. It won't come like this. Yeah, go ahead. Go ahead. These are the XPT
so
files.
like we had to open those. It's a like is it like some app that Pinnacle 21 software or like
Yes,
like
Pinnacle 21 software. I don't know if I when I was in off when I was working in an organization on these things, I had the access of Pinnacle 21 there. But I don't know if I can upload it randomly. I have this Pinnacle 21 community, but I don't know whether it will open. I have not tried it. This is the problem. Like personal, it probably needs some kind of a it probably needs some kind of a license or something based on which you can open it. But the working you can see on YouTube, how Pinnacle 21 works. It will show you step by step how it actually works.
okay. So suppose we have to again upload this XPT file in the SAS environment, so we cannot open, but like how to access them?
That is called a proc import is there. Proc
Okay.
import functionality. Remember, did you do proc import in SAS?
Yes. Yes.
Ah,
Yes. Yeah.
that proc import. And similarly, when you export the XPT, you have proc export.
Okay.
So you have, if I want to proc export and this out of here, then I'll use the proc export functionality. And if I
Okay.
have to import this XPT, I can use proc import. Proc import, I can import whatever. Like I can export a CSV, I can export Excel. Remember, I can export an XPT, I can do whatever. See, the understanding the concepts which I explained you here, right? These concepts are important. I'll tell you one thing very frankly. This SAS programming, all these things which we are doing right now, this you can do it from Google as well. Okay, you can just go, I mean, SAS program syntax to import, to export for proc beans. You will get it in Google as well. But what is the concept behind it? How this, why are we doing this SDTMs or why, well, not SDTMs? Why are we doing these tables and listings and figures? What do you think? To make you understand what exactly they mean. What is the concept? How a clinical trial actually works. That's why I spoke to you about the CDM first. This understanding is important. Coding anyone can do. Anyone can do means if someone has a knowledge about it, if you get stuck somewhere, how do seniors handle this? If they get stuck somewhere, they do Stack Overflow, they go to Kaggle, they do Google search, they go to ChatGPT, and that is where the syntax you will understand these ones. But why are we doing this? That's what I'm focusing on. That's why when we started the course, I had shown you the entire clinical trial workflow, what we are doing, what not, how we will be doing it, what are the like what is clinical trials, before clinical SAS, what are the processes, and this is something which we discussed about. This is the project. This is the pro forma. This will help. This is something which the people will be looking into, and some basic level of coding. Because this is something which you will go and get it from the Google as well, right? What exactly is happening is like how to constrict a number in SAS for if I need one decimal figure. You will get this. You will get all these things, and you can take it. Important part is to get the concepts understand. What is SAP? What are the adverse events which are there? What are the clinical events? That's why I'm asking you to read the protocol once. Go to the protocol, read it. That is where you'll get a lot of understanding of what's exactly happening. And also the CDISC IG, SDTM IG, which I had given you. Go through that. It's self-explanatory. It's not difficult. If you just read through it, you will get an understanding. You'll be more confident during the interview because you have the knowledge of the domain. That is what is required. And it is best that you have the knowledge of the domain along with this technicality. Got it? Priyanka Shagufta, any questions?
Yes.
Yes.
No, Archit, no question.
No, no. So one more question, Archit. What is the ELT?
What is ELT means?
Like I like there is some short form I heard like ELTs. I don't know what that. I thought of asking you.
I know ETL.
Okay,
Extract,
ETL. Sorry. Yeah.
extract, extract, transform, load.
Okay.
You extract data from somewhere using any technology, be it SAS, be it Python. You transform it based on certain conditions, and you load it into a separate environment. But why do we need that?
Yeah. That's it.
Huh. Okay. So I will one second. I'll do one thing. I'll copy paste this here. Okay. So let's quickly go through this table once. Okay. Uh, Let me explain you the concept of baseline a little bit. What is baseline? So baseline is basically uh, think of a person, think of a patient who has come to for a clinical trial on the first visit. Suppose V1, okay, and his pulse is, suppose I'm giving some random values. Don't go by a little value. His pulse is suppose 120. Okay, same person comes to the same person comes to the clinical trial, gives a second visit on V2. Now it's seen that his pulse remains same. Again, that person comes to V1. So uh, this is only for pulse, okay? Not pulse. Suppose BP, sorry. Okay. That person comes to V3 and we see his pulse now 140. Now the person takes a drug, okay? After taking the drug, the person again comes to the visit. Now it's V4. His BP is 110. Again, that person comes on another visit. Now his BP is 160. And again, that person comes for a last visit. This person is, we can see, 130. Now tell me, just before taking the drug, when is the when is the value just before taking the drug? What was the what was the BP of the person?
140.
140. Okay, 140. Correct. So baseline is something which we normally define as the value which is measured for any particular vital signs or lab just before taking the study drug. Okay, so suppose this is the baseline value. Okay, So tell me one thing, if this is the baseline value, so let me put the baseline value here as well. So we have considered this as the baseline value. So this will be 140, this will be 140, this will be 140. Okay. Now what will be the change from the baseline? What will be the change here?
Nothing.
Nothing.
Minus 30. Minus 30.
Exactly. See this value. What is change from baseline? Baseline was 140. After taking the drug, is there a change? Yes,
Yes.
it's been 110. So what is the change? 30. Let's take absolute values. What is the change? 20. What is the change here? 10. This is basically the situation which we would be looking into in this table. So for tafenoquine, if you see treatment, what are the visits which are there? So suppose this is the baseline visit. So we will only have observed value here. So see, if we have like this is the treatment. So these are different. I mean, these are for separate parameters. Suppose this parameter is for BP. Suppose we take BP. So for drugs, how many people are there who has this value on this screening visit? Observed in mean, median, what would be it? And what are the change from baseline? And that change from baseline, what is their N, mean, median, SD, mean, max? This is what we are trying to establish here, but just for safety population. Now, if I go to the directly to the data set, okay. Suppose let's go to the data set once, ADVS. So we have observed value is eval. Let's go to the eval. So see, these are the visits. Okay. These are all the parameters for diastolic, systolic. You can see day 1, day 2, day 3, day 4. All these things you can see. And these are the eval. Okay. Analysis value. That's what they have asked us to take eval. And if you see change from baseline as well, it's there. Go to the right. CHG. Do we have CHG? So if you see something, see diastolic pressure, blood pressure. This is the heart rate beat. Okay. So if you see diastolic blood pressure for this person, this is 002. Okay. Let's concentrate on that. 102, 002. If you go to the right, see that person 102, 002 has come for 2 visits, screening and day 14. Okay. First visit and last visit probably he has come. So he has been taken 2 values. What are those 2 values? One is 89 and one is 70. Clear? Clear. 89. For first day he has been given 89 and the second day he's been, I mean, it was measured 70 for his blood pressure. Now, what is the baseline value? If you see base, it's showing 89. See? Because when is the first one? It's 89. It is showing, see, for both the results, it's showing 89. Now see the change. First one will be null. Why first one will be null?
89, 89.
Absolutely, absolutely. So if I go here, if this is if I give you a change from baseline, this is really 140, 140, it will be 0. But what is the next one? Minus 19. Do you see that? Because 89, 70 minus 89 is minus 9. Next is what we have. We have 86, 79. But it is for heart rate. It's for the same patient, but the parameter is different. 86 and 79. Okay. Now what will be the baseline? If you see base, it's 86. So we have null here. We have minus 7 here. So we have to take this for different parameters and see for how many different patients. So what is the, so how, what all we have got? Let's come back to this one. So what all we have got? Number of people for this screening visit and day 14. So it is basically screening visit and day 14. In between, if there are other visits, what visits are there basically? Or one second, let me see which visits they have taken here. No, they have randomly, they have not taken any specific visit. For every visit, they have tried to see. Because every visit, there would be a baseline. I mean, there would be a change from baseline, correct? So starting from here, if you see, this is the screening. So basically, if you see that this is the first visit, so you will not have any change here. But from day 1, day 2, day 3, day 4, day 5, you will have an observed value as well as change from baseline. So how many people have this change from baseline? All those people, what is the mean for, what is the mean change from baseline for heart rate, for pulse, for oxygen saturation? We will have all this. What is the median? What is the standard deviation? Everything will be. Understood? Yes or no?
Yes. So just one question. What is this a day minus 4 to or minus 1 means is like the screening days, like 4 days is going on in
No,
the.
not 4 days going on. 4 days before the screening day. Suppose screening day is considered as 0. So the
Okay.
patient has started coming 4 days before. That's why screening day minus 4 to minus. This is confusing. I understand. This is something this can be made very clear. Like the first day when the person visits, it can be treated as screening 1, then screening 2, then screening 3. Then when he gets when he starts to get the drug, it can be marked as day 1, day 2. But that is something which is not done. So it's basically the parameter is taken as day 0. 4 days before you get screening minus 4 to minus 1, like minus 4, minus 4, minus 3, minus 2, minus 1, like that. Here we have given like screening minus, so the screening is 0. From the 4 days before screening till screening, anywhere the patient has come, we can get this. So if you see, I mean, anywhere the patient has come to the hospital when we have given the vital signs, that will be considered that value. So suppose this is visit 1, we can consider it as screening minus 3. We can consider this as screening minus 2. We can consider this as screening minus 1. And this drug day, we can consider it as day 1. Okay. This is then you can see day 2, day 3, day 4. Got it?
Yeah.
Okay. Now, let's look into another scenario. Okay. So the person has come. So the person who is being treated has come on the first day visit, has come on the second day visit, did not come on the Thursday visit, has taken the drug, and then has continued. So in this case, what will be the baseline?
You mean to say post baseline or like what?
What will be the baseline value which we should consider?
So last one only, right? The
Where
last one,
last one, which is what?
the baseline.
Baseline. Then what will be the value?
Like V3, we don't, so like 120.
Correct. Absolutely. We don't have we don't have this. Correct. So can we have this as a baseline? The immediate value before taking the drug when the patient has come to the clinical trial. So we should be considering 120. This will be 120. But this will change to 10, 40, and 10. Got it? Absolutely
Yes.
clear.
Yes, yeah.
This is the concept of baseline and change from baseline. So I am not explaining this again by this one. You can just see it. I will share this code with you. This is not that difficult. You will get to know how it is done. It's very simple. You can go through it. But this has been done for different parameters actually. So I'll show you how the data comes in basically. Where using this macro, where equal to. So where did we give something? Where equal to, okay, this one. Parameter. Let's execute this and check. You can basically look into this table with, I mean table at home as well. But how this table comes up. You see, for systolic blood pressure, we have 43 people. So for systolic blood pressure, there are 43 people who has given his blood pressure on the screening day and this one. So if you see one thing very clearly, Priyanka and Shagufta, for screening it's the baseline. That's why you've only got observed wala part. Do you get this part change from baseline? Because it will always be zero, right? In the change,
Yes.
this will always be zero because you are considering which visit? You are considering the baseline visit for all the participants. That will obviously be zero because we don't have value. But the moment you come to day 14, you have this value. So 39 people are there. So for on the C, on baseline, so what is derived from this table? So on the screening visit day, on the first visit, 43 people showed up. All these are the mean, median, SD, mean, max. But on the last day, only 39 people have shown up. And this is the value. And you are correct. We are not considering the absolute value. We are considering negatives as well. So you are correct, Priyanka. Okay. We should not consider the absolute value. If it's negative, we consider negative. Again, see for placebo. This is for the drug. Again, for placebo, if you see, 43 people showed up during the screening. So that is why they understand, boss, what happened to these 4 people? Where are they? So they are not there. So at the start of the study, everyone came. At the end, hardly people have come. We only gathered data from 39 people. This is
So
only
here is one question, Arjit. So this is the last day data, or like day 1 to day 14 data?
no, just day 14 data.
Just only day 14 data.
Just day 14 data. Because next one you see.
Oh, okay, yeah.
Got it?
Yeah.
This is temperature. Then we have heart rate. Again, heart rate, we have 43 people, 39 people. Only these two. Like heart rate was taken on this first screening and at the end of the study. Then what we have? Okay, we have only till this one, heart rate and temperature. We don't have anything else. We have acha, 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 acha. Got it, got it, got it. Where param, if we have just given 1, 2, 3, you can give your param 1, 2, 3, 4, 5, 6, whatever number if you give, you will get more results. You'll get everything, oxystat. Well, there are lots, right? If you go here, if you see here, there are a lot of data that you will be able to see. ABBS, if you go, we have just done param 1, 2, 3. So you can give 15. No, you can give diastolic blood pressure, oxygen saturation. There are lots. You can give everything, temperature, weight, diastolic, everything you should give and check. And there are two aspects to it. The upper portion of the code is dealing with the macro. The lower portion is dealing with the normal. Okay, so that is the reason. Check both. Okay, check both and practice both.
So is there any change in the program, or are we doing
Yes,
the
obviously,
same?
same program is there, but one is macro. So we have given everything within the macro, and we have just have to run here. If we have to give 1, 2, 3, parameter. If for parameter we have given 1, 2, 3, you can give more and you will get more results. But here, if you we have to give in parameters what? If I go down, go down. If I go down, okay. Here we have not given any difference. We have given all the parameters here. Here we have in the end we have given all the parameters. I mean the code which is this part. So we have probably taken all the parameters. Class by param, and we have taken all the parameters data would come in. Then we have done merge. This is a little bit.
Can you show the like first part, like where are you filtering in the macro? I did not get those.
This is the where we are filtering. First, we are doing proc means data, where ampersand where. This is ampersand where. So where we are doing what? If you go here, ampersand where is SAFFL equal to Y, safety flag, and param n equal to 1, 2, 3. So what do we have? Param n is 1, 2, 3. What do we have? Param n in 1, 2, 3. Check, check. 1, 3 is heartbeat. So we got heartbeat. 1 is what? 2 is what? 2 is temperature. We got 2 temperature, and 1 is systolic blood pressure. Now you can check, you can use 5, 6, 7, whatever. So this is the decising factor here while we are doing proc means. Okay, where class param, paramein, all these we have taken by treatment and a visit and everything variable, and we have given this. Again, we have done proc means for the change. See, PDVS ampersand where variable is changed, and at the top variable is given. Because we need two, right? We need two differences. One we need the observed, and one we need the change from baseline. Got it? Here also, if you see down, which is the simple without the macro, instead of means we have given here proc summary. Same concept. Proc summary, variable eval. If you go down, proc summary, this is change. Both are the same, proc summary and proc means. We can use to calculate this one. Tell me, Priyanka, any questions? Where are you hesitating somewhere? Priyanka Shagufta, tell me.
So we can do this with the proc means also, right? Like this is the
Absolutely. But have you done proc summary before?
um, no.
No. So this is where you can learn proc summary. It's basically the same thing. So when you execute this code, you will easily understand. You will easily get it that okay, these two are the same thing. So in an interview question, if they ask you, have you used proc means proc summary, you can say yeah, both have the same functionality. And if you find any difference, major, you say that. But both are same. That's why this purposely I did the proc summary with I did the proc means with macro and proc summary in this normal format. Got it? Tell me any questions if you have. I'm stopping the recording.